Okay, good to see you again. Let's start our last three phases of mitosis, the first of which involves metaphase. If you look at this diagram, some really interesting things are happening. You can see that the chromatids are beginning to form across the center of the cell. And the spindle fibers that have attached between both centriole at both ends of the pole are beginning to run through those chromatids at their central location point, which were the centromeres. So we can summarize that up in bubble A, knowing that chromatids line up across the center of the cell, and the chromatids connect to spindle fibers at the centromere. And that main part here in the middle is often referred to as the metaphase plate, and this is going to be instrumental in the actual division of these chromatids in the next phase. Alright, let's take a look at the next step, known as anaphase. Some interesting things are happening here. If you can see, right where those chromatids lined along the middle of the cell, they're beginning to break apart. So the duplicate copies that were formed on the centromere are being pulled towards the opposite poles. So let's start with bubble number 1 to summarize. Centromeres of each chromatid pair begin to separate. And what you begin to see here involves bubble 2. Daughter pairs of chromatids are pulled towards the opposite poles. And just like the diagram indicates, what you start to get are your daughter chromatids are moving towards each opposite pole and the centrioles at those poles. They're moving along the microtubules that were formed along the centriole pairs 2 phases ago. And what is unique about this step can be summarized in box 4 as well, that now you start to see the cell elongate, meaning that it's increasing the distance between both poles. So it's increasing in length, indicating that it's very close to separating and mitosis is close to being complete. The final stage we're going to talk about in mitosis involves telophase. This is kind of unique. You see some things happening here that are interesting. One of the main components associated with this phase involves the nuclear membrane reforming. It's reforming as a way to organize the two sets of chromosomes, the two sets of organelles, the two sets of nuclei that are beginning to develop and almost close to dividing apart. So the nuclear membrane reforms, and at this point the nuclei enlarges and the chromatids uncoil. Number 2, we know that a cleavage furrow exists, and this cleavage furrow is right in the middle of where that cell is located, right at the point where dividing is going to occur. And at this point there's a thinning that exists. The nuclear membrane and the plasma membrane that once existed as a whole is beginning to thin, indicating that separation is going to be in the near future. So we can summarize that up in bubble number 2. The cleavage furrow begins to develop. This is a thinning process for cell division. Bubble 3 summarizes the next step. We know that the daughter nuclei begin to form. So the formation of the nuclei, and with the chromatids uncoiling, the genetic information, the DNA and things like that, that process is going to be complete, forming a nucleus that is complete and ready to function. All right. At this point, step 4, we can see that 2 new cells are almost completely organized into 2 separate cells, and the next step would involve the process of interphasing, returning, and preparation for cell division to begin again. If you have any questions on this, let me know prior to class or during class. We'll definitely spend the first few moments of class to review, and we'll go from there.